Hey everyone, this is Miss Hunt, and this is Honors Biology. We're week 12. All right, let's click into our Google Classroom. And for week 12, I've got my lesson plans on here, and then a lot of good stuff. So let's go into those lesson plans real quick. Some of you guys are doing a great job in getting all that work turned in on time, and some of us are still struggling. If you have problems with USA Test Prep, you need to let me know Monday through Friday, 7:45 until 3:45, and I will help you out. Asking me Friday night at 9 o'clock doesn't work. So make sure you guys are getting me those questions within the timely manner. All right, today we're going to go through 9.1 of our new notes of mitosis, and we're going to go through the study guide and finish that 9.1 study guide. All right, we're beginning a brand new unit, and we're talking about the cell cycle. We've talked about how DNA reproduces. We've talked about DNA building protein.
We are ready for bus drivers. Thank you.
But now we're moving towards the good stuff. We are talking about how the cell reproduces and all the fun parts of that reproduction. As you can see, I've got on here the rest of mitosis, including a lab on Thursday. I hope you're here and you're ready to do the lab on Thursday. Friday, this is your last Friday off of class, off of school. You still have an assessment on USA Test Prep to take, so make sure you get that done. We have Thanksgiving break, and then we come back and we're back to school on that Friday. So we will continue taking tests on USA Test Prep on Fridays. So it won't be that much different, except you're taking it at school instead of being at home. All right, and of course, I still have a few who are staying at home online, and some of you guys are doing great, and some of you are really struggling. I can only tell you that your grade will improve when you come back to school. The more you are here, the better your scores will be. It's just you go over more and we talk more and we have a better opportunity to learn. All right, here is the meiosis week, and then we are we finish reproduction for sexual and asexual reproduction. And then the week before Christmas break. I know it'll happen so fast. We're doing a review for our midterm, and we're taking our midterm. So it'll be all on USA Test Prep. It'll be with the USA Test Prep questions that you've seen before, and that'll be it. And we'll be done with with semester 1. So I'm really excited about that. So this will be coming up. So make sure you're keeping up with the lesson plan. If you're at home or quarantine, every day do that day's lesson so that you don't get behind. And I shouldn't be getting stuff turned in at 2 a.m. Sunday morning, Monday morning, when things need to be done. Don't wait to that late. Don't turn stuff in that late. Don't stay up that late. It's not healthy for you. All right, so let's go in here. We did chapter 9 notes, the cell cycle, today. I also have a printed version. If you want to take notes on here, I highly recommend that you do that. It helps you learn being able to write those notes out. Okay, so a brand new unit of the cell cycle. This is very much putting things in order, okay, as best I can tell you. And keeping it up in order. Okay, we're just going to do section 1 today. So our notes begin with, we've seen this slide before. This is that surface to volume ratio. Remember we did the diffusion lab where we had 3 different sizes of cubes, a small, medium, and a large? Remember which one that everything diffused very quickly through? That was the small one. And the larger one took a long time. So we're talking about why do cells need to make a new copy? Why make a new cell? And it's basically because the cells can't just grow bigger. A bigger person doesn't have bigger cells. They just have more cells inside of them. As a cell grows, the volume increases much more rapidly than the surface area. The cell might have difficulty supplying nutrients and expelling waste and expelling enough waste product. Right? Transport of substances. Transport moves by diffusion or by motor protein. Pause. Diffusion. Do you remember what type of transport that was? Passive or active? Hopefully you guessed passive transport. Remember, passive doesn't require energy. Our water diffuses this way, glucose, oxygen go by diffusion. Very simple through that plasma membrane. Okay, also you could go through protein, motor proteins. Do you remember they look like the big proteins that are in the phospholipid bilayer where things move in and out? I hope so. All right, diffusion over long distances is slow and inefficient. Small cells maintain more efficient transport systems. So this slide's about why do we have to make a new cell? Because the cells can't grow too big. Small cells are better. Why are small cells better? Transport substances more efficient. Okay, so that was transport. This one is about communication. The cells needed for signaling proteins to move through the cell also limits the size. Cell size affects the ability of a cell to communicate instructions for cellular function. So let's go back to the last unit. Ring, 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 nucleus, we need to build this protein. Transcription happens, we build the mRNA. mRNA leaves, travels to the ribosome. The ribosome does translation, builds the protein. Protein gets packaged up in the ER, sent to the Golgi. But what if the ER was far away from the Golgi and the message got lost? It was hard to communicate the signals. It's hard to even reach the nucleus to tell it to make the protein to begin with. So a small cell is better. Why? Cell communication, transport. Okay? All right, therefore, we do the cell cycle. Cell division prevents the cell from becoming too large. It also is the way to cells reproduce so that you grow and heal from certain injuries. Cell reproduce by a cycle called cell cycle. All right, let's go back to this slide right here. Oops, sorry. Okay, so this is 4N to our notes. I want you to pause and think, what was the main point of these 4? Hopefully you have that small cells are better. Better for communication, better for transporting of information and signals, better for diffusion. Small cells are better. Why can't we get big cells? They're inefficient. Small cells are better. Okay, so we're going to go through the cell cycle. This is very much, like I said, putting things in sequential order. You have to memorize the order. All right, the first growth of the cell cycle is interphase. I'm making you a picture like I usually do. Okay, so the 3 parts to the cell cycle are interphase, mitosis, cytokinesis. We want to memorize those in order. Interphase, mitosis, cytokinesis. All right, interphase, the stage in which the cell spends most of its time, it's like 67% of its time is in interphase, and it carries out its normal functions. Let's remind ourselves, what's the functions of the cell? They're supposed to make ATP in that mitochondria. They're supposed to store food in the vacuole. They're supposed to build proteins, doing protein synthesis. They're supposed to just be a cell. They make enzymes that digest things. So that's interphase. Most of the cell's time is in interphase. Mitosis is the division of the nucleus. Mitosis makes a new nucleus. Everything else happens in interphase, but mitosis is just nuclear division, just the nucleus. And finally, my last word is cytokinesis. Okay, let's all say cytokinesis. All right, that should be some Latin in here. Cyte, C-Y-T, you've heard it before. Cyte means cell, like the cytoplasm was the cell's fluid. Cyte means cell. Kinesis reminds you of telekinesis, or maybe kinesthetic, or maybe Potential and kinetic energy. Do you remember? It should be motion. Kinetic energy is of movement or motion. Kinesis is moving. So this is where the cell moves. So I use my hands a lot in this unit because hands are really easy to manipulate. All right, so let's pretend that this is one cell. And let's do interphase. My cell should be growing bigger in interphase. It's just being a cell, doing its job. All right, then mitosis. The nucleus makes a copy. That's called mitosis. It pinches apart, cytokinesis. Now I have two cells, and they're identical, supposed to be identical, to the original cell. So again, my one cell grows bigger. The nucleus makes a copy. That's called mitosis. Pinches apart, cytokinesis. I recommend doing it with your own hands. Pinching apart, cytokinesis. Okay? All right, we're not done yet. There are phases inside of interphase. So in interphase, we have G1. You can say growth 1. Some people say gap 1. G1, S, and G2. Okay? So here we go. G1, S, and G2. And this is what's also written up on my board in my classroom. Okay? Interphase, G1, S, and G2. So what's happening? All right, in G1, where you can see a lot of time is spent. All right, first of all, what is this circle thing? Kids were always getting confused with the circle. The circle represents time, the lifespan of a cell, the cell cycle. All right, what you should see is that between G1, S, and G2, which is all interphase, the cell spends most of its time doing this. Okay? Mitosis is relatively short, and then very short is cytokinesis. Cytokinesis. All right, in G1, the cell's growing and it's being a normal cell. In the S phase, we are synthesizing the DNA. Let's stop and think. What does that mean, synthesize the DNA? All right, you should remember the 4 enzymes that were involved in DNA replication. Who comes through and unwinds the DNA, unzips it? Did you guess helicase? That's good. Okay, once it's unzipped and coming apart, somebody waves a primer and says, start here. Who says start here? Remember, they write it in a different language? The RNA primase, good. The RNA primer comes in and puts down the RNA primase. And he writes an RNA language. Remember, that means ribose, sugar, and uracil. He's just giving a starting point. Who comes in with the hard hat and starts building? The DNA polymerase starts building the new strand of DNA. Remember, the leading strand goes towards the 3' and he could take off and do that really easily. The lagging strand had to work backwards in those fragments. What are those fragments called? Okazaki fragments. Very good. Okay. The last enzyme comes in and he starts to glue the Okazaki fragments together. Do you remember him? Ligase. Very good. All right, so all of that, and you're left with 2 pieces of DNA, half old and half new. What do we call that? Where we've held some back. Semi-conservative replication. All right, all of that from that last unit is right here in this S phase. DNA replication. All right, then we go to gap 2, where the cell prepares for mitosis. Finally, mitosis is when the nucleus divides, and then we have cytokinesis. It pinches apart. Okay? All right, the third stage, gap 2, the cell prepares for division of the nucleus. All right, we copied this slide just so that we could get to the next picture because we wanted to go through the phases of mitosis. So there's a lot of vocabulary here that we're going to have to get to next time. All right, cells chromatin, so this is prophase, the first phase of mitosis. Chromatin tightens and sister chromatids attach at the centromere. And the spindle fibers form in the cytoplasm. There's a lot of vocabulary here. We're going to go over it tomorrow. Okay? All right, what I want you to know today is putting these in order. Prophase, metaphase, anaphase, and telophase. We call it PMAT. PMAT is mitosis. Mitosis is PMAT. Okay. All right, so there's mitosis. Mitosis is PMAT. PMAT is mitosis. Mitosis is P. Oh my gosh. Hold on just a second. My son's getting tired. I don't think I've ever messed that up. But I did. Let me do that again. Okay, let's try it again. All right, mitosis is PMAT. PMAT is mitosis. P-M-A-T. Prophase, metaphase, anaphase, telophase. P-M-A-T. Prophase, metaphase, anaphase, telophase. Don't tell anybody I messed up. Phase. All right, so let's look at our slide here. Prophase, metaphase, anaphase, telophase. So tomorrow, we're going to talk about what is happening, what are these things, and all the vocabulary that goes to it. All right, what I want you to know is this. Can you put these in order? Just sequential order. Interphase, G1, S, and G2. You should know what they are. You should know the 3 parts of the cell cycle: interphase, mitosis, cytokinesis. And the phases of mitosis, PMAT, if you can get that right and in order. Today we partnered up. We had a reteaching of explaining what interphase was, and then another person explaining what the phases were in order. Remember, the more you talk about it, and I know talking about things at home is hard, but the more you talk about it, the better it will be. All right, after this, you need to do the study guide, okay? Just section 1 today, 9.1. Okay? Just 9.1. Here you're drawing that circle and labeling the parts of the cell cycle, okay? And you should be able to answer these questions on here. All right, that's today. Tomorrow we're doing this one tomorrow. We're sequencing DNA, okay? We're looking at mitosis. We're going to look at modeling it with string. So I would recommend you getting something, pipe cleaners, string, even a piece of paper to help you with the modeling of mitosis. We're also going to be creating a flip book. Now at home, this is going to be harder to create the flip book because you need paper to flip. If you don't have paper to print this out, you can always email me and come pick it up at the attendance office. You can definitely fill this out on the text PDF, but you can't flip that way. So it is fun to make a little flip book of the mitosis, and then we cut it out and go, and it like flips through. So we can kind of see it that way, and that works pretty well. The lab doing that at home is going to be hard. I have put some reference pictures in here for you. We go back in here. Here's some reference pictures to look at. And this kind of gets you looking at, like, what is this? How do I know what it is? So this is basically what we'll look at through the microscope in the prepared slide. Okay, things like this. So you have to look at it and then be able to say, what phase is this in? That should be metaphase. This is anaphase. They're being pulled apart. So you're looking at the slides and the microscopes and trying to identify what phase they are. Hopefully there's enough reference materials on here for you to look at the cells and be able to recognize them. This standard says we're supposed to develop and use models to explain the role of cell reproduction. So you need to be able to draw and look at models of pictures and what this is. So that's what you need to be looking at and working on at home is, do I know what this is, what phase it's in? Do I know when that phase occurs? And of course, there's a lot of vocabulary that goes with it. All right. I hope you guys are doing good, and let me know if you have any questions.